Hello, we're nearly to the end of Module 1 and time for just a quick review of everything, well, of some of the things in Module 1. It's a heavy module, lots of things to learn and remember. Please be sure that you're using the general discussion board so that I can answer your questions or your colleagues can, and everybody can benefit from the answers. So just a quick review, pharmacodynamics, what medications do to the body. So for instance, a medication can alter receptor function. Consider beta blockers. They block the ability of norepinephrine, epinephrine to actually occupy that beta receptor, resulting in no action from that receptor. Pharmacokinetics, what the body does to the drugs. And so this is things like metabolism by CYP450, absorption, distribution, binding, things like changes in GI function, liver or kidney function. These are all things that can change pharmacokinetics and therefore the amount of drug that's present in the body and whether or not it's effective or over-effective or toxic, etc. These are all determined by pharmacokinetics and pharmacodynamics.
Now, when we think about pharmacokinetics, we got to think about the CYP450 system. It's a profoundly important thing when we're considering the medications that we're going to be prescribing because they metabolize so many drugs that we use. So when you think about CYP450, you have to remember inducers and inhibitors. So the medication that's actually being metabolized by the enzyme is a substrate. For instance, some of the antipsychotics are substrates of CYP1A2. Okay, so they're metabolized by that drug. Now, inducers increase the amount of enzyme available. If I increase the amount of enzyme, I increase its action. So if I increase its action, what does that mean? It means more of the drug is being broken down because that's what all of this is about. When we metabolize drugs, we're changing them from the form that's used in the body to the form that's excreted by whatever means, right? So inducers increase the amount, and there's an inverse relationship. So if I increase action, I decrease levels of the drug. So an example of an inducer, cigarette smoking is a potent inducer of CYP1A2, which can lead to reduced levels of many of the antipsychotics. One exception, risperidone. Now, to inhibit something is to slow it or block its action. So if I give somebody an inhibitor of a CYP450 enzyme, I slow the metabolism of that drug. I slow the breakdown, which means more drug is being left behind. So levels will rise. Okay? So that inverse relationship, a slower metabolism is a higher level in the bloodstream. An example of this, grapefruit juice. Potent inhibitor of CYP3A4 and 3A5. People who take this and they're taking benzodiazepines like diazepam or alprazolam may have increased levels of that benzo, which can lead to a possibility of increased side effects, which nobody wants. Now, first pass metabolism is an interesting phenomenon. And let's see if I can get this video to do what I want it to do. Yes. Wait. Yeah. I'm already in Chrome and I just updated it. Well, ladies and gentlemen, I think we'll skip that for today and I'll attach the link to
Well, so much for my tech working the way I want it to today.
So, let's talk about the prefrontal cortex. The prefrontal cortex actually has three segments: the dorsolateral, which sits uppermost in that prefrontal cortex, right? So prefrontal cortex is, of course, just behind our frontal bone. Ventrolateral and orbitofrontal, which is really just behind the eyes. So the dorsolateral prefrontal cortex is the topmost part and is considered to have management of cognitive processes like planning, cognitive flexibility, working memory. This area specializes in problem solving and how to direct and maintain attention, right? So it helps to prioritize. When we're focused on what we are doing, our working memory is engaged with the dorsolateral prefrontal cortex, connecting with the hippocampus, which remember is responsible for memory, for the retrieval and consolidation of long-term memories. A dysfunction in this area, the dorsolateral prefrontal cortex, can lead to problems with working memory, processing in the hippocampus, which helps us to consolidate memories, and in long-term memory, as well as with some of the integration of expression of emotions, particularly verbal expression. Such memory deficits we see with things like PTSD, we see with attention deficit disorder, although less so than PTSD. We can also see with dorsolateral prefrontal cortex problems with things like spontaneity, problems with affect, where they appear very flat rather than negative or sad or anything like that. We also see the attention deficit, so the inability to maintain attention to actually follow a task through to completion. In OCD, we sometimes see the dorsolateral prefrontal cortex play a role in strengthening those attention skills to the point where they're unable to change to something else, where it becomes an obsessive thought. It sometimes is also able to help us to break that circuit and give the orbitofrontal cortex a chance to inhibit the runaway activation of places like the amygdala, where some of those where those compulsions will come from, right? Because we're afraid, someone with OCD is afraid that if they don't follow through with this action, that there's going to be consequences. And so this is where some of that interruption of that can come from. There's differences between right and left hemisphere with all of these. The left dorsolateral prefrontal cortex is more likely to be associated with approach behaviors, right? So where we actually go into whatever it is that we're maybe afraid of. And the right is more likely to be avoidant behaviors, where we run from something that we don't want to have to deal with. The orbitofrontal cortex is involved in cognitive processing and decision making, but it has a really close connection with the limbic system. And it's particularly associated with decisions based on emotional information, right? It plays a role in social attachments, emotional regulation. It's kind of, if you think about it this way, it's kind of a place where sensory and emotional information come together from external and internal to create perceptions and guide how we interpret interactions. It plays an important role in things like understanding a joke, right? It pulls together the kind of factual data. It pulls together things like tone of voice, facial expressions, etc., so that we can actually interpret what's going on with the world. And it modulates our behavior accordingly. So when there's dysfunction here, we don't have that same modulation. And we see that sometimes with borderline personality disorders. It does have hemispheric differences, just like we saw with the dorsolateral prefrontal cortex. The left is more associated with positive emotions, and the right with more negative emotions. Now, the ventral medial prefrontal cortex, we don't see as often described in a lot of the literature, but it does play a really important part. And it is part of the prefrontal cortex that helps us to make decisions based on big picture. So it connects with temporal lobe, amygdala, the ventral segmental areas. It sends and receives a lot of information from a lot of different areas of the brain, including the amygdala. It plays an important role along with the orbital frontal cortex in regulating emotions, especially when we're dealing with social situations. It's also vital for decision making and ability to learn from our mistakes, right? It gives us our ability to make judgments and to allow our emotions to assist in decision making through mediation of the ventral medial prefrontal cortex. Also associated with things like courage, through the suppression of things like fear. It is also responsible for suppressing some negative emotions like shame or guilt, which maybe some people need more of. Now, the nucleus accumbens, tiny little thing, which is thought to mediate behaviors, including especially awards and motivation. It's in the basal forebrain. It's a major structure of the ventral striatum associated with the limbic system and regulates limbic functions like motivation, affect, and reward. The most widely recognized function is its role in the reward circuits of the brain. The nucleus accumbens is a huge component of a dopamine pathway called the mesolimbic pathway. And the mesolimbic pathway is stimulated during rewarding experiences. It's one of the things that we see fire up when people are using drugs that they're addicted to, right? When they're getting that high. We also see it kind of light up when you're eating a favorite food or doing something that you enjoy. This is the area, part of the ventral tegmental area, that is activated. Now, the raphe nuclei. Raphe means a line or a ridge that separates two symmetrical parts of the body. And this is pretty, it's appropriate because the raphe nuclei sit right around the midline of the brainstem. And these are composed of cells that are part of the reticular formation, but they're responsible for the serotonergic pathways. So the neurons that produce serotonin originate in the raphe nuclei. So it's responsible for a lot of things. The raphe nuclei and the serotonergic pathways that erupt from it project into almost every area of the brain. It's responsible for mood, emotion, responses to stress, cognition, sleep, movement, sexual function. So very important. But this is where this area here is where the serotonergic neurons originate and spread out to the rest of the brain. The locus coeruleus is the primary site of norepinephrine producing neurons. So this nucleus sends norepinephrine and noradrenergic neurons throughout the brain, again, all throughout the brain, amygdala, hippocampus, cerebellum, spinal cord, basal ganglia, the whole kit and caboodle. So norepinephrine has projections into so many different places that it is very strongly linked with many processes, including things like attention, vigilance, arousal during stress. It has an effect on sleep. So lots of different ways, but the noradrenergic neurons originate from this area of the brain, the locus coeruleus. The amygdala is a fascinating place. The amygdala, you'll often hear it called our fear center, and this is true. But really, the amygdala manages information about many strong emotions. It processes information coming from prefrontal and temporal cortices and the hypothalamus. It's, it has pathways that terminate in the septum, the hypothalamus, the thalamic nucleus. It has connections to the basal ganglia, the ventral pallidum, the ventral striatum. So all of these circuits really come down to the processing by the amygdala of information related to any strong emotion. So we typically hear fear, but anxiety, aggression, emotional memory, social cognition, all of these things are processed in the amygdala, and then it sends information to other areas of the brain, like the prefrontal cortex, as we talked about earlier, which is trying to prioritize decision making based on interpretations of the data that it's receiving from places like the amygdala. It's also involved with things like appetite for food, sex, and drugs. It has a role in memory when the amygdala is strongly activated, like when somebody has a very big traumatic event. The amygdala can disrupt memory processing, leading to a lack of consolidation of memory. There are very specific functions like responses to stress, crucial roles like responses to fearful stimuli, drug-related stimuli, and an interesting area called the mid nucleus of the striae terminalis, which is also involved in anxiety and stress. Little bean-shaped thing, lots of different functions, lots of disruptions that can occur from there as well. Now, I wanted to kind of bring back and just kind of remind you about CertNet and DAT. These are the transports for dopamine, norepinephrine, and serotonin, respectively, right? So the effect of these is the movement of dopamine, norepinephrine, or serotonin from the synaptic cleft into the synapse, right? So they carry the molecules from the cleft into the cells. When we block them, we do the reverse. When we block CertNet or DAT, we actually end up with whichever neurotransmitter it's responsible for becoming more numerous, the levels of that neurotransmitter increasing in the synaptic cleft. So it has the effect of blocking reuptake. Okay. Now, one of the things to remember with this, these require active transport. In other words, the level of neurotransmitter in the synaptic cleft, there's no osmosis. It doesn't just move based on concentration. It requires the body to expend energy in the form of ATP to move a sodium-potassium pump to exchange, to pull that neurotransmitter across that semiconductor. Now, the agonist-antagonist spectrum is one that you really need to be comfortable with, right? This is something that you're going to see over and over and over again. And many of the concepts that we're talking about in this first module, you will see on certification. They're not looking for you to just learn this stuff and forget it. You will be asked questions about the brain, about neuron function, about agonist spectrum, about the cytochrome P450. These are all things that you will continue to need throughout the program and to get through certification. And I use this stuff every day in my practice. Okay? So the agonist spectrum reaches from agonists, which actually occupy the receptor and activate them, through antagonists, which block the action of the agonist. Now, a lot of people think that An antagonist does the opposite, but that's not necessarily true. It just blocks anything from happening, right? An inverse agonist would be the opposite, right? Where if a receptor, just as a oversimplification, if a receptor, when it is stimulated, agonist, makes you happy, the antagonist just blocks that happy from happening. An inverse agonist, if it occupies that receptor, makes you unhappy. So the opposite. So an agonist makes it go. An antagonist just makes nothing happen. An inverse agonist does the exact opposite of what that receptor would ordinarily do. Now, partial agonists are also in there. Partial agonists kind of give you something between full agonism and antagonism. So it's kind of like a partial response. And then you have modulators. And sometimes you'll hear partial agonists called referred to or described in a way that says they're modulating, and they can. A modulator will take, for instance, the drug cariprazine, Vraylar, is a D3, D2 partial agonist and modulator. So what it does is in areas of the brain with too much dopamine creating positive symptoms, it reduces the dopamine. And in the areas of the brain with not enough dopamine, where we have negative symptoms, it increases dopaminergic function. So kind of review the agonist spectrum and be comfortable with it, because you'll need it again and again. Now, the HPA axis is discussed a lot, and some of our research into antidepressants, especially, is using things like corticotropin-releasing hormone or ACTH. So it's important not just for understanding the hormonal impacts. It's also important because some of our future medications are going to actually be coming from this concept, right? From this system. So the HPA axis is part of our stress response. Okay, so in response to any stressor, and remember, these stressors can be good, they can be bad, they could be anything. In response, the hypothalamus signals for an increase in norepinephrine levels, and it does this by releasing corticotropin-releasing hormone to the anterior pituitary. Now, the anterior pituitary releases adrenal corticotropic hormone, ACTH, which circulates and hits receptors in the adrenal cortex, which release cortisol. And we know that cortisol is one of our stress hormones. When cortisol is released into the bloodstream, it increases wakefulness. It increases our fuel for the stress response, right? Because cortisol, like prednisone, increases gluconeogenesis and glycogenolysis in the liver in response to a stressor, so that we can get ready to run, part of the stress response, right? So it gets us ready for running with more fuel to the muscle, so it increases glucose. It also inhibits things that are not necessary right now, like anybody who's ever worked in OB, which no, never. If somebody has a lot of stress, if they're really anxious, it actually slows their labor. That's cortisol saying, no time to have a baby while we're running from the big bear, right? We also don't have time to stop and urinate or defecate, right? Those things will actually help whatever's pursuing us to find us. So the stress response inhibits some of those things, right? So it's essential. The whole HPA axis is a huge, very important part of our day-to-day life. It is required in order for us to survive. However, remember that prednisone and cortisol also suppress immune function, right? And so long-term activation of the HPA axis can cause a lot of physical and psychiatric problems. So they have problems with immune response. Type 2 diabetes, obesity, cardiovascular disease are higher in people who have a prolonged stress response. It also has, cortisol specifically, has detrimental effects on things like memory, cognition, and high cortisol has been implicated in mood disorders like depression. We see this especially in studies that have looked at young children who experience profound stressors, who their HPA axis remains hyperactive. The weird thing is that HPA axis in hyperactive mode eventually kind of poops out, and we end up with a less perky stress response over the long haul, which means they're less able to deal with stress, they're more likely to have depression, and lots of other areas of problems with cognition can occur. So that's why there's so much research right now on HPA axis as it relates to things like mood and anxiety and other facets of psychiatric medicine. Very exciting stuff, by the way. Now, DNA methylation. DNA methylation is an epigenetic change. So remember, epigenetics are things that occur to affect the expression of our genes from environmental, developmental, pharmacological, age, diet, what have you. So these are things that are occurring from outside the cell that change how DNA is expressed. Okay? So this can cause a gene, for instance, to be read or not read, so expressed or silenced. And that occurs as a process called methylation or acetylation or a couple of others that change the structure of the chromatin. So remember, the chromatin is part of what helps to bind the DNA together in that helix. Okay? So when there is methylation of DNA, what happens is it takes that spiral and it squishes it together. Okay? Now, if I take a spiral, think about a slinky. Okay? If your slinky is sitting at rest, you've got that normal spiral, you can see all of the different areas. You can poke your fingers in at different parts of the spiral. But if I take that slinky and I go like this, and I compress it, the inside of that spiral is no longer visible or accessible. The same thing happens with DNA methylation. So when RNA is sidling up to the DNA to replicate and make enzymes, that squished part of the chromosome is no longer readable and is therefore silenced. Those enzymes are not produced. This leads to some of the diseases that we see as a result of things like stress or environment or development or what have you. So it has the effect of essentially inactivating the genes that are in that segment that's been compressed, right? Now, demethylation or deacetylation can occur, and the result then is the opposite, right? So if I demethylate DNA, and this can happen from a number of different things, it actually spreads that helix, that spiral, back out again so that those genes are in effect turned back on again and expressed. Okay? So that's the end of my module 1 brief review. My plan is to do this for each of the different module segments so that you have a chance to kind of to get a little extra information. Again, make sure you're using that discussion board. I would really prefer that you don't send me information about or questions about content by email, because then if I get an email from each of you every week and I answer once instead of 12 times, that would be brilliant. Not that I don't not that I mind getting on email with you, but let's save the email for things that you don't want to share, that are personal, about grades or that kind of thing, or something going on with you that you need an extension. Use that common discussion board for asking questions so that everybody can benefit from the answers. Okay? Looking forward to more this week. I will talk to you all throughout the modules. Looking forward to it. See you next week.